Right now, if your last meal was 24 hours ago, the cells lining your small intestine are not resting, they are primed. The LGR5 positive stem cells, the ones responsible for rebuilding your entire gut lining every 4 to 5 days, are sitting at elevated readiness, waiting for the signal to proliferate. That signal is not hunger. It is not time. It is the specific chemical composition of whatever you put in your mouth first. Your gut does not care that you fasted. It cares what you hand it when the fast ends. And it will respond to that first bite faster and more dramatically than at any other moment in your day. This is the part of OMAD that almost nobody talks about. Not the fast itself, the landing. 24 hours of precise metabolic work, and then a single meal that either honors the state your body built or erases it before the benefits finish accumulating. Before we get into what that means for your first meal, quick question when you break your fast. Do you eat immediately when hunger hits, or do you wait until a specific time? Drop your answer in the comments below. Here is the simple version first. After 24 hours without food, your body is running on a completely different fuel program. Insulin is near the floor. Fat is feeding your heart and muscles. Your liver is manufacturing glucose from scratch to keep your brain supplied. And inside your cells, a molecular cleanup process has been running for hours, dismantling damaged components and recycling the debris. The first bite ends all of that instantly. The question is what you hand the system to work with when it switches back on. Now here is what is actually happening at the cellular level, because the mechanism is the thing that makes the answer obvious. By hour 24 of a fast mTOR1 mechanistic target of rapamycin complex 1, the master switch your cells use to decide between repair mode and growth mode has been suppressed for the better part of a day. Low insulin means the PI3K, AKT signaling pathway that would normally activate it is quiet. AMPK, the energy scarcity sensor inside each cell, has been holding it down. Autophagy, the cellular maintenance process that breaks down damaged proteins and worn-out organelles and feeds the raw materials back into the system, has been running at elevated activity since roughly hour 12. Your cells, in the most literal sense, have been cleaning house. The moment food arrives, mTOR1 reactivates. MIT research tracking this transition found that blood glucose, which had dropped by around 50% during a 24-hour fast, returned to baseline within 30 minutes of the first refeed. That is not a gradual recovery. That is a threshold crossing. And mTOR1 follows glucose up almost immediately, switching the cellular machinery from breakdown to construction, from cleanup to growth. Here is what the MIT researchers also found, and it is the part that reframes the whole story of fasting. The gut regeneration benefits the surge in intestinal stem cell proliferation. The tissue repair, the cellular rebuilding, did not peak during the fast itself. They peaked during refeeding. The fast created the conditions. The first meal triggered the harvest, which makes the first bite not just the end of the fast, but the most biologically active moment in the entire 24-hour cycle. Most people treat it like a reward. Their body treats it like an instruction. What this actually feels like is the moment the maintenance crew packs up and the building opens for business again, except the building has spent a day quietly replacing its own plumbing, and what arrives through the front door next is either going to stress test the new work or let it settle properly. Your digestive system has been standing down. Gastric acid secretion is reduced after an extended fast. Digestive enzyme output from the pancreas is reduced. The migrating motor complex, the wave of electrical activity that sweeps through your small intestine roughly every 90 minutes during fasting, clearing residue and maintaining gut motility, has been doing a different job than processing a full meal. Your gut is primed for repair. It is not primed for a large glucose load arriving all at once. This is where what you eat first becomes the entire conversation. A fast carbohydrate rice, bread, anything that breaks down quickly to glucose hits an insulin system that has been suppressed for nearly a day and triggers a release proportionally large to the size of the signal. The dual-wave insulin response that normally handles a carbohydrate meal fires hard. A stored bolus from the beta cells of the pancreatic islets first. Then a second wave of newly synthesized insulin 15 to 20 minutes later. Gluconeogenesis. The process your liver has been using to manufacture glucose from amino acids and glycerol and recycled lactate since hour 8 shuts down almost immediately. Lipolysis, the enzymatic dismantling of fat stores that has been feeding your muscles and heart, is suppressed by the same insulin signal. Beta-hydroxybutyrate, the primary ketone body circulating through the fasted state, is cleared from the bloodstream within 60 minutes. 24 hours of metabolic architecture, gone in under an hour, because of what arrived first. Put simply, a carbohydrate-first meal ends the fasted metabolic state in one move. That is not inherently wrong. You are going to eat, and the fasted state was always going to end, but it ends abruptly, before the fuel-switching machinery has had any chance to hand off gradually. The consequence is a glucose load landing in a system that was running on a completely different program a few minutes earlier. Your body handles it. It has always handled it. It is just doing considerably more work than it needed to. Amino acids behave differently. When protein arrives first before significant carbohydrate, it triggers an insulin response, but a slower and more modulated one. The incretins GLP-1 and GIP rise from the intestinal wall in response to amino acids, amplifying insulin release, but the glucose signal that would drive the large second-wave response is not there yet. Leucine, one of the branched-chain amino acids, abundant in eggs, fish, and most animal proteins, directly activates mTOR through a parallel pathway that does not require insulin as an intermediary. The research here is specific. Leucine activates mTOR via the RAG-GTPase complex on the lysosomal surface, independently of the insulin, PI3K pathway that glucose triggers, which means protein first initiates the anabolic switch, the signal to start rebuilding without the sharp glucose spike that carbohydrate first produces. We are getting into the mechanism here because it is the only way to understand why the sequence matters, not just what you eat, but what arrives first. Think of it this way. Your liver after a fast is a furnace that has been running on a slow, controlled burn for 24 hours. A carbohydrate-first meal is throwing a full bucket of accelerant in at once. A protein-first meal is adding a measured fuel, one that keeps the furnace running, starts the transition, and lets the system shift gear rather than lurch through it. The third element is fat. Dietary fat triggers the smallest and slowest insulin response of the three macronutrients. It also directly stimulates cholecystokinin CCK, the gut hormone released from the duodenum that slows gastric emptying and signals satiety to the hypothalamus. A small amount of fat before or alongside the first protein source slows the transit of everything that follows, reducing the rate at which amino acids and any subsequent carbohydrate reach the bloodstream. The mechanism responds to sequencing. Fat, then protein, then carbohydrate, in that order, is not a wellness protocol. It is the shape of the transition the mechanism is already trying to make, helped along. Think of gastric emptying the way a queue management system works. Everything you eat joins a line before it enters the small intestine. Fat moves to the front and installs a slow gate. What arrives through that gate, amino acids, then glucose does so in an orderly procession rather than all at once. The gate is not magic. It is cholecystokinin doing a job it was always designed to do, triggered by the fat you put in first. The honest gap in this is worth naming. Most of the clean human data on post-fast refeeding sequencing comes from shorter, overnight fasts, 12 to 14 hours, not 24-hour fasts. The mechanistic logic holds. Lower insulin, more pronounced mTOR, one suppression, larger contrast between the fasted and fed states. But the precise magnitude of the effect at 24 hours versus 12 hours has not been tested head-to-head in large human trials. The mechanism points clearly in one direction. The dose-response data for fasting duration specifically is still being assembled. Anyone telling you they have the exact optimal sequence time to the minute for a 24-hour fast is working ahead of what the evidence can currently support. What is not in dispute is the refeeding transition itself. A 2022 study tracking the circulating metabolome during supervised fasting and refeeding in humans found that the transition from fasted to fed state was among the most metabolically active windows measured with amino acids, fatty acids, and ketone bodies all shifting within the first hour of refeeding in ways that differ substantially, depending on what the first meal contained. The fed state is not a return to baseline. It is a distinct metabolic event, and the first meal writes its character. Over days and weeks, consistently managing this transition, fat first, then protein, carbohydrate later in the meal, maintains the enzymatic efficiency of the fat-burning pathway. The fat oxidation enzymes that upregulated during the fast do not simply switch off the moment you eat. They are sensitive to the insulin signal that follows. Keep that signal gradual and the pathway stays primed. Slam it with glucose, and the pathway gets the message that it is no longer needed. Over months, the insulin sensitivity data is consistent enough to take seriously, though the magnitude of the effect varies considerably between individuals, which is something most summaries of that research quietly omit. Your body spent 24 hours building something. The first bite is not a reward for surviving it. It is the moment the construction either continues or gets cleared to make way for something else entirely. Tomorrow, before you eat, try shifting the order. A small amount of fat, first a handful of nuts, a drizzle of olive oil over whatever you are about to cook, then protein, then carbohydrate. At the end of the meal, 30 seconds of sequencing. The mechanism will run the rest. Now that you understand how the first bite either extends or ends the fasted state, there is a second half to this story, and it lives in what happens when the transition goes wrong. Like, subscribe, and stay tuned for the next video to find out about the hormonal sequence that follows a large carbohydrate-first meal after an extended fast, why it produces the energy crash most OMAD eaters recognize, and what the mechanism tells you about how to avoid it.